Okay, so welcome to the makeup video for your February 22nd BISC 101 lecture. Again, sorry for not being there today, but rest assured that the doctors checked out Katie and say that she's going to be totally fine. So thank you for your well wishes and for your patience. So continuing on where we left off, so we were talking about how a neuron works, and we already spent quite a bit of time talking about how the membrane gets charged with a negative membrane potential, measuring inside versus outside, thanks to the action of sodium-potassium pumps and sodium and potassium leak channels. And we were talking about how when other nerves send signals to the nerve we're focusing on, it can cause this negative membrane potential to go up or go down with these graded potentials. We'll talk more in detail about these kinds of connections, these synapses between neurons a little later, but what I wanted to focus on first was what happens when enough of these excitatory potentials bump up the membrane potential until it reaches the threshold, and that causes an action potential to fire. All right. So at this point, enough excitatory signals came in from these other neurons and caused these postsynaptic potentials, these little graded potentials, to add up enough to reach the threshold, and that caused the axon hillock, having reached threshold, to fire an action potential, this great dramatic increase and decrease in membrane potential. All right, so just to review kind of the simplified versions of what happens after this first action potential is initiated, is this kind of big influx of positive charges ends up spreading a little bit further down the axon and depolarizes the next part of the axon. So you can think about this entire axon as being in resting potential, so just sitting there at the resting potential waiting to be triggered. And once an action potential gets triggered here, that positive charge spreads down a little bit and causes the next segment of the axon to reach threshold, and that causes another action potential to fire, and the positive charges from that action potential again spread further down, make the next segment reach threshold, and that fires another action potential. And so you can kind of imagine this resting potential as we have kind of a little bit of like a big line of resting babies, and if you were to initiate a big reaction, a big cry session, a tantrum from one of those babies, that effect is going to cause a chain reaction of babies waking up and crying. So in a similar way as the action potentials, basically have a chain reaction of activations going down, wow, 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 all the way to the end, until the signal reaches these axon termini, and we'll talk about what happens after that, right? So once the signal reaches axon termini, it's going to be passed on to other cells through synapses, okay, which we'll discuss later. But for now that you understand the simplified version of how an action potential gets initiated, thanks to these excitatory signals from other cells, how that signal gets spread in a chain reaction of action potentials all the way to the end of the axon. All right, so let's look at how this works. So having done the simplified version, let's look at kind of the detailed version of how action potentials are actually made. Right, so what makes the axon hillock and the rest of the axon act so differently from the dendrites in the cell body is that, remember the cell body and the dendrites have ligand-gated channels which open to let a little bit of sodium in or potassium out, and that causes these small changes in membrane potential. Well, the kinds of channels found along the axon are quite different. They are voltage-gated. So we have voltage-gated sodium channels in pink and voltage-gated potassium channels in purple here in the axon. And when this membrane, right, so these are studied all throughout the axon here. So when this axon membrane is at the resting potential, in the axon as well as the axon hillock as well, the voltage-gated channels stay closed. All right, so when you're at the resting potential, sodium channels closed, you see nothing's going through, potassium channels closed, and so there's nothing changing, the resting potential just stays there. Now, when the membrane potential, though, thanks to these excitatory signals, when the membrane potential rises up and hits the threshold, though, so basically at the axon hillock, the membrane potential is no longer at the resting potential of -70, is now risen up to something like -50, where the threshold is. All right, so once membrane potential reaches threshold, then things start to happen. The sodium channel, sensing that change in voltage, will now snap open, and that causes sodium ions to be able to flood in. So remember, there's lots of sodium ions on the outside. They move in by diffusion through these voltage-gated sodium channels. And think about it, if you have a whole bunch of positive sodium ions coming into the cell, that's going to change the voltage inside the membrane, right? So rather than being at negative 50, it's going to shoot up high, get depolarized, meaning it's losing its polarization, actually go past 0 all the way to positive 40 millivolts. All right, so in this first phase, the sodium channels snap open, lots of sodium floods in, and we lose that negative charge, so much so that we go past 0 and go all the way up to positive 40 millivolts. So in phase 2 of the action potential, right, sensing that the membrane potential now is shot up to this much more positive membrane potential, the voltage-gated potassium channels respond to that different membrane potential, and they open up too now. So soon afterwards, the voltage-gated potassium channels open, and potassium ions start to flood out of the cell. Because remember, potassium's concentration gradient is flowing in the opposite direction, out of the cell. There's lots of potassium inside wanting to leave. So now, since we opened up the potassium channels and potassium can now leave the cell, we now have lots of positive charges leaving the cell, and as positive stuff leaves, the inside becomes more and more negative again. So now we're going to repolarize our membrane. So whereas we lost that negative charge before, we're going to gain it right back again by letting positive charges out, letting the potassium out of the cell. Okay, so that's the repolarization phase, where the membrane goes back to negative 70, to the resting potential. Now, phase 3 comes immediately after that because the potassium channels stay open so long that some extra potassium ions go out, and that actually allows the membrane to go even more negative than normal. So this is hyperpolarization. It's not just repolarized, it's polarized past the resting potential. All right, and this is important because it makes it harder for another action potential to be fired very soon afterwards in the same segment of the axon. All right, but that's basically the entire shape of the action potential. We have depolarization caused by opening of the voltage-gated sodium channels, letting sodium ions flood in and make everything positive on the inside. Then potassium ion channels open and let a bunch of positive potassium ions leave, and that causes it to go back to negative and even get hyperpolarized for a bit. And then notice that over time, after being hyperpolarized, the membrane does go back to its normal resting potential because of that dynamic equilibrium that's always there between the sodium-potassium pump and the potassium leach channels as well. So between the pump and the channels, things do always equal out back at the resting potential. Okay. Here's another figure that's similar, but it actually shows you the effect of opening the sodium channels in pink, right? So when the sodium channels opens, that's what causes the membrane potential to shoot up very high, right? But as the sodium channels close and the potassium channel opens up more and more potassium floods out, then that causes the membrane potential to drop back down and even get hyperpolarized before the potassium channels close and things can slowly come back up to resting potential. Okay. So now that you kind of understand how an action potential is made and the types of voltage-gated sodium and potassium channels involved, we can look at another scientific thinking question, which actually is a very good practice exam question. When I say practice exam question, I don't mean that I'm going to give you the same question in the exam, right? The point is to make you think about situations. But this is the kind of thinking and problem solving that we expect you to be able to do in your final. All right, so I'm basically going to give you a situation, ask you to predict the data, predict what the graph will look like, all right? So this here is a picture of what a normal action potential looks like, and I want you to predict what would happen to this action potential, this change in membrane potential, if we blocked the voltage-gated potassium channels, okay? So let's say we put a drug into the body or onto an excised nerve, and we basically block all of the potassium channels, and then we stick an electrode inside of and outside of the axon membrane, and we're measuring what happens to the action potential, what does it look like then. So what I want you to do is, in a minute, I want you to pause the video and think about what might happen when you block the potassium channels. So draw a graph of what the data might look like if you were to block the potassium channels. And once you've done that, and I want you to think about how that will affect the organism itself. Okay, so how does this blocking of the potassium channel affect the membrane potential for an action potential, and also what would happen to the organism as a whole. Okay, so pause the video now and answer that question. Okay, welcome back. So hopefully you are actually pausing the video to use your worksheets and write down your answers and commit to them before you see the answers, because that is a very big part of your learning, to teach yourself to look at a new situation and figure out for yourself before you spoil the answer with the next slide. So please do pause before you continue. Okay, so in case you were stuck about how the heck to draw the figure, I'm going to give you kind of 4 common options. So when we ask students to do this in class, these are some of the graphs that they drew. So I want you to take a look at these 4 graphs and see which one looks like the one you drew, or which one of these 4 you think is the most correct. Right? So pause the video and choose one and write that down. Commit to that answer on your worksheet. Okay, can you pause now? Okay, welcome back. So I would say that the best graph would be C. All right, and if you disagree, if you think you have a better graph, please do come see me in office hours, and I'd be happy to discuss it with you. But I would say that the membrane, so I would say C is correct because we still have our voltage-gated sodium channels, and so if you trigger an action potential in the neuron, the sodium channels will still open and allow sodium to flood in, and that's what's going to make the membrane potential shoot up very high, up to positive 40 millivolts again. But at that point, normally, the potassium channels will open, and that's what causes the potential to go back down and repolarize, right? But without the potassium channels, that's not going to happen quickly. But I would say that this is not correct. I would not expect that the membrane potential will stay where it is, because remember, we still have the sodium-potassium pump working, which is making things negative on the inside. We also have our potassium leak channels allowing potassium to leak out, which again makes the inside more negative. So the action of those 2 types of membrane pumps and membrane proteins are going to bring the membrane potential back to the resting potential once again. Okay, so that's why I think C is the best answer. All right. The second question was, what do you think would happen to the organism itself? Well, if you ask me, I think that the organism is going to die pretty darn fast, right? Because if this happens, if this drug is concentrated enough that it's affecting every nerve in the body, every neuron in the body, not only is the brain not going to be working, also the neurons involved in stimulating the heartbeat are not going to be working, as well as for your breathing as well, which is controlled by your nerves. Those nerves will not be firing. So think about it. If your heart's not beating and you're not breathing, your heart is not beating and you're not breathing, you're not going to survive very long. All right. So let's look in detail at how an action potential, now that you know how it's made, how it gets spread down the axon. All right. So let's say that this part of the axon is undergoing an action potential. So it's at the very beginning stages in phase 1. The voltage-gated sodium channels are open, and sodium is flooding into the axon. Right, so lots of positive charges coming in, which is why you see that instead of being negatively charged inside like the resting potential should be, it's actually positively charged at positive 40 millivolts. All right, so as these sodium ions flood in, all these positive charges, positive ions that are already in the axon, kind of get pushed away from this region. Right, think about it. If you're in a positive ion that was in this region to begin with, and a whole bunch of positive things come in, like charges don't like each other. Like charges repel, and so that's going to push other positive charges further down the axon, and that movement of positive ions into the next region basically depolarizes the next segment until that segment reaches threshold as well, and boom, we have another action potential happening here. We open the voltage-gated sodium channels. As those positive ions flood in, they spread the positive charges further into the next segment, causing that to depolarize to the threshold, and once again we'd have an action potential in the next segment, and that's kind of how the chain reaction spreads down. You can also see that once the next segment has been triggered and it's depolarizing, the second phase of the action potential, the repolarization, is already happening here. All right. So overall, we're talking about the spreading of these positive charges caused by the influx of sodium to the next part of the axon, and that depolarizes that segment enough to start a new action potential, and again, this basically repeats all the way down the axon. Okay, so as a scientific thinking question again now, so what do you think would happen? All right, so this is an interesting situation. So let's say I took a neuron that's completely at rest, I pulled off all the other neurons and nothing's going to activate it, right? And I went and used a very fine needle to inject a whole bunch of sodium ions into the very middle of this axon. All right, so here are your options. So read this carefully, commit to the answer that you think is right on your worksheet, and then restart the video to see what I think is correct. Okay, so pause the video now. Okay, welcome back. So I'd say the correct answer is C. All right, so if you think about it, if you inject a bunch of positive sodium ions into the middle of the axon, right? So if you have the whole axon membrane at the resting potential and you add a bunch of positive charges, you're going to depolarize this pretty quickly so that it gets to the threshold. And at that point, all the voltage-gated sodium channels here will snap open and start doing an action potential. So a whole bunch of sodium, extra sodium comes in from those voltage-gated sodium channels. So that positive charge is going to spread outwards in both directions, right? If you're a positive ion, you're not staying where there's lots of positive around. You're going to be drawn over to the negative over here and the negative over here. And that causes both of these segments to get depolarized enough to hit the threshold. And now we have action potential on the right, action potential on the left, and that's going to keep spreading to the right and to the left. So action potential is spread in both directions. So considering the neuron itself is capable of sending the action potentials in either direction or both directions, why is it then the action potential normally only go from left to right, from the cell body and dendrites towards the axon termini? Well, there's a few reasons for this. Right? You can pause the video and think about it first if you like, but then to move on. One reason why the signal always goes from dendrites towards axon termini, or not towards dendrites, from the axon hillock to the axon termini, is that neurons only receive signals on this end, right? So the axon itself and the axon termini, they don't receive any signals from other neurons, so there's no reason why you would initiate a signal at this end or here. You're always starting the signals here at the axon hillock. Okay, so depolarization always starts on one end and kind of chain reactions its way down. That's one reason. Another reason is that, remember, after an action potential happens, right? So let's say that you depolarized, you cause an action potential here, and you would think that these positive charges want to go in both directions, but as these positive charges come down this way, that part of the segment is actually being repolarized or hyperpolarized, so it's going to be harder to bring this to threshold and make it fire backwards. Okay, so when an action potential is just completed or in the middle of completing, it's harder to trigger a new one, so it's less likely that this new action potential will cause action potentials backwards. But the most important reason is this. This is a voltage-gated sodium channel, and you'll see in a second when I start this video that after it opens to allow these little green sodium ions to come into the cell, there's a flappy thing here that actually closes the channel, right? So that even if the channel is open, nothing can flow through. All right, so you can take a look here. Okay, so you see sodium going in, and then that flap closes. All right, so you can. This video will be part of the slides that I post, so feel free to check this out when you're reviewing. But here it is again. Sodium ions flood in, and soon afterwards, that part of the protein closes things off, so that even if you were to depolarize this previous section up to threshold, nothing would come in. You wouldn't allow any sodium ions in, therefore you would never get the action potential bouncing back and going backwards. All right, so these are kind of reasons why you would not get a new action potential triggered going in the opposite direction of what's normal. Okay. All right, if you want to review how all of this works, there's a really cool simulation that can kind of help you see the big picture all at once with some real numbers, right? So you can click on this link when you're reviewing with the PowerPoints, and I think the best way to see the most detail and to learn the most from this is to first go and click all of these show boxes. So show all the ions, show the charges, show the concentrations, show the potential charge, right? And then also zoom in a bit. So notice that I zoomed in about halfway, right? And then at that point, you can go ahead and hit the stimulate button, and that's going to cause an action potential to flow down this axon, and when it reaches this part of the membrane, you can see what actually happens. Okay, so a few things I want you to kind of pay attention to when you're playing with this simulation. First thing is that when you have an action potential, it only uses up a tiny bit of each ion's concentration gradient. So you can see the concentration gradients here. So the sodium concentration is way higher outside, 145 millimolar, and way smaller inside, just 10 millimolar. Okay. You also see the opposite for potassium. It's 140 millimolar inside, so 140 huge on the inside, and only 4 on the outside. All right. So this is maintained normally by your sodium-potassium pumps and your lead channels, and you'll see that happening in the simulation. And when you fire an action potential, though, you don't lose all of that 145. You only lose a little bit, and it shows up on this side. Okay, and then slowly the sodium-potassium pumps will pump it back. Okay, Okay, so the important thing about this is that since you only use up a little bit of this concentration gradient, it means that after a neuron fires, you don't have to wait for things to recharge before you can fire again. You can pretty much fire as soon as those sodium channels are no longer blocked. Okay. You can also notice how after an action potential, these concentration gradients get gradually restored back to where you started from, thanks to the sodium-potassium pumps and the leak channels. So after each action potential, those pumps and channels are working away, restoring your resting potential. Okay. You can also see this little blocky thing after sodium channels open as well. Okay. So go ahead and learn from this. This is a very useful model for you to learn from. Okay. So let's talk about how action potentials can travel faster down an axon, because we mentioned earlier that when you cover sections of the axon with myelin sheaths, and you do that by basically having a Schwann cell, which is a specific kind of glial cell, like a non-neuron nervous system cell, it basically grows and wraps itself around and around and around the axon, makes a thicker and thicker kind of multi-layered wrapping or insulation around the axon. Okay, so when you add a myelin sheath, so when you have these little wrapped sections of the axon, the signal can go through that axon. The action potential can move through that axon about 300 times faster. Okay, up to 300 times faster. So that makes a big difference if you're trying to signal, like, say, your foot to contract and move, if you can get that signal there 300 times faster. All right, so the reason why the action potentials can travel faster is because these action potentials can actually jump from one node of Ranvier, meaning this, so we have the Schwann cells covering up and insulating this segment. The node of Ranvier is these uncovered sections, these unshielded sections in between. Okay, so action potential, when it happens here, can basically influence the membrane channels here to cause an action potential to fire here, and then it jumps to this one and so forth. Okay, so here's one action potential happening. The positive charges push their way down, depolarize this segment, causes action potential, because you need to expose these membranes channels to let the action potential happen, and then again that spreads further down and causes another action potential. Okay, so let's talk about how that happens. How does the action potential manage to jump from node to node instead of spreading down the whole membrane? Right? Well, normally, right, when you have an action potential, these positive charges do kind of push and leak their way further down the axon. But as those positive ions are shoving each other down the axon, a lot of them also just leak back out of the axon, and the effect for later parts of the segment are not as strong. Right? So pretty much the sodium ions and the positive ions only push down enough to depolarize the very next part of the membrane before too many positive ions are leaked out, and we have to wait for a new action potential before the positive charge can be further spread down. Okay, so normally without the myelin sheath, only nearby regions are triggered, and so you get action potential here, spread a little bit, action potential here, spread a little bit, action potential here. But when you add a myelin sheath, though, you're basically wrapping this insulating layer, this kind of barrier to charge movement, so that when these positive charges come in, they can spread down without leaking out as much. So the effect of these positive charges moving can actually spread way further down the axon and cause an action potential to fire way down here instead. Okay, so fewer ions leak through the membrane when it's wrapped with the Schwann cell with a myelin sheath, and therefore you're able to shift enough positive ions down to the next node of Ranvier to depolarize it, and that causes another action potential here. And once again, these positive charges are trapped in this insulated segment and then spreads down and causes another action potential further down here, and that just continues all the way down the axon until we reach the termini. Okay, so one thing to notice is some people think that when there's an action potential and sodium ions flood in, you have to wait for those sodium ions to, well, push each other and also diffuse their way down and actually reach the next node of Ranvier. But that's not actually true. You don't need the sodium ions themselves to move to the next node. Okay, so when sodium ions flood in, they don't actually travel to the next node. The way you can think of it is more like this. You know that this axon is already filled with positive charges, right? Like the potassium ions that are in high concentration inside, as well as other positively charged molecules as well, or ions as well. So if you suddenly shove in a bunch of positive charges, those positive charges will shove each other further down the axon. So the way you can think of it is this. So think of the positive charges as a bunch of people on a very packed train, okay? Say like in Japan where they really get packed full. All right, so all of these charges, all of these people are trying to keep an equal distance away from each other, give each other as much space as possible, even though it's really packed. So that's what the resting axon looks like. Now let's say that you open some sodium channels and you shove a whole bunch more sodium ions into the train, right? Now that's going to cause all of these other people that are already in the train to shove further down the train, right? Getting as much space as they can again. And so the effect of these people coming in are going to be felt much further down the train, right? Maybe way down here where they're flooding back out again. All right, so that's kind of what's happening here is the sodium ions come in and they shove the next positive charges, they shove each other, and everybody just shifts down a little bit. And those positive charges shifting into the segment depolarizes it, and that causes a new action potential. All right, so if you ever go to Japan and stand in a subway, you'll know exactly what I mean. Okay, so moving on then. So in this way, whether it's with myelinated axon or not, the action potential in one way or another is going to spread all the way down to the axon terminal. So now what? What happens after that? Well, our neuron here is now going to signal other neurons using their axon termini. So the way that one neuron signals another is via synapses. So a synapse is what we call the connection between one neuron and another. Okay, so there's two different kinds of synapses, so two different ways in which two neurons can meet and affect each other. Okay, now an electrical synapse and a chemical synapse. All right, now electrical synapse is quite a bit simpler in a way. Basically, in an electrical synapse, we have gap junctions, which are these specialized membrane proteins that actually just connect the cytoplasm of this neuron with the cytoplasm of this neuron and allow ions to flow back and forth pretty freely. All right, so what that means is that as an action potential spreads down this membrane and reaches the very end of this first presynaptic cell, the ions flooding in, like the sodium ions, for example, that flood into this cell, basically just spread their way through the gap junction, or push other positive ions through the gap junction, and that basically will for sure depolarize the next membrane enough to activate a second action potential. All right, so this is an all or nothing thing. As long as these two cells are connected with an electrical synapse and an action potential comes in, the action potential is pretty much just going to spread right to the next one and continue signaling. All right, so it's an all or nothing kind of response. Now, a chemical synapse does something a bit more indirect in that when an action potential reaches the axon termini in this presynaptic neuron here, instead of directly signaling the next cell to an action potential automatically, what it does instead is it releases a specific neurotransmitter, a specific chemical, into this gap here, and that chemical binds to ion channels, which open and either allow ions in or out, and that produces graded potentials. Okay, so this neurotransmitter acts as kind of an intermediate messenger between one neuron and the next, and this is how we generate those graded potentials that we were talking about before. All right, so I'll talk about in detail how these synapses work in a minute, but first I just want you to kind of take a pause and think about what the pros and cons of each type of synapse might be. All right, so pause the video now and use your worksheet to brainstorm some ideas. Okay, welcome back. So electrical synapses, one of the benefits of it is that it's very fast and very consistent. So it doesn't need to wait for this neurotransmitter to diffuse across and open channels and stuff like that. Pretty much the ions just directly push each other further into the next cell and depolarize it. The action potential goes through pretty much without a pause. So the potential is triggered right away, and it's triggered pretty much every time, right? There's an all or nothing thing. Now, chemical synapses are different in that the effect of the presynaptic cell, the neuron that's bringing the signal to the synapse, it has a more graded effect, a more subtle effect on the postsynaptic cell. So this makes it so that instead of every time this neuron comes in, it signals the next one to fire for sure, it gives you those little bumps up and bumps down in memory potential in this next cell instead. So that allows you to actually integrate the input from a bunch of different chemical synapses. Okay, so this basically just fires no matter what. This one can fire or not depending on what all the other chemical synapses nearby might be doing. All right. It also allows for inhibitory signals as well, whereas this one doesn't. This one just says go, go, go. This one says, some of them can say, okay, maybe fire soon. The other one might say, don't fire. Be less likely to fire instead. Okay. Besides allowing for these more subtle and integratable signals for the postsynaptic cell. Another thing that's cool about this is that this is where kind of learning and memory happen, as far as we can figure out that by changing how this subtle signal works, so by making it stronger or weaker or breaking these synapses, that's how we can learn new things and memorize things and remember and recall them. All right, so chemical synapses are very important. So that's what we're going to take a close look at them now. So terminology-wise, again, we have two cells synapsing with each other right here. The synapses are here, and here's a zoom in of that. But we just want to show you that this whole cell that comes before the synapse, we call the presynaptic neuron, which makes sense. It's the neuron that comes before the synapse, and the one that's being signaled is the postsynaptic neuron. So if we're talking about the membranes on either side of the synapse, this is now the presynaptic membrane, and this is the postsynaptic membrane. Okay, so let's go through this step by step. So step one, the action potential is spreading down, down, down this axon all the way to this terminus. Okay, so this is the action potential arriving, and that basically continues to depolarize and depolarize the membrane all the way down. And in response to that action potential coming by, there are some calcium channels here at this axon terminus that opens up, and there is now an influx of calcium into the cell. So as those calcium channels open, since the concentration gradient for calcium is to have lots of calcium outside and very little inside, as soon as those channels open, calcium starts to flood into the cell. Okay. Now I left a blank here because I wanted you to pause and think about what kind of calcium channel this is. Is this a ligand-gated channel that's waiting for a chemical to open them, or is it a voltage-gated one? So pause and think about that for a sec. Okay, welcome back. So since these calcium channels are opening in response to the action potential coming in, then you should have guessed that these are voltage-gated calcium channels. They're responding to that depolarization caused by the action potential. And in response to that, they say, oh, okay, we're depolarizing. This is a signal, time for us to open, right? So they respond to voltage, okay? So once these voltage-gated calcium channels open, calcium floods in. That flooding in of calcium is another messenger that tells these synaptic vesicles, meaning these little kind of container spheres here, these membrane-bound spheres. These are synaptic vesicles that will now fuse with the presynaptic membrane, and as they fuse with it and flip inside out, all of those neurotransmitters are going to be released into the synaptic cleft. The synaptic cleft being basically the gap between these two membranes, right? The little separation between the two nerve cells. Okay, so now we've released the neurotransmitter, and as the neurotransmitters then diffuse across this very small gap, which doesn't take very long, and then they can bind to proteins on the postsynaptic membrane. So notice that as they bind to the protein, these might be ligand-gated sodium channels that will open up and let some sodium in, and that will depolarize the membrane a bit. Okay, so overall, we've had the action potential come in, we've released the neurotransmitter diffuses across, binds to the membrane protein of the postsynaptic cell, postsynaptic membrane, and that causes a change in the membrane potential. Okay, so it basically causes this membrane to become either a little more negative or a little less negative. Okay, so this change in the membrane potential of the postsynaptic membrane, so this change in voltage in this cell here, we call that a postsynaptic potential, meaning a change in the membrane potential of this postsynaptic membrane. Okay, so taking a closer look at this membrane protein here that's binding to the neurotransmitter and letting some stuff come in, I want you to take a look at this and think about what kind of channel that might be, right? So there's a bunch of different types of channels it might be. So think about it and pause the video now. Okay, welcome back. I want to give you a little bit of a hint before I reveal the answer. So here's a hint. So a ligand is basically a small molecule that binds to protein, right? So, and this can apply to a bunch of different scenarios, but generally speaking, a ligand is a small molecule that binds to protein and can change the protein in a way that kind of turns it on or turns it off. Okay, so think about your options again and pause the video. Okay, welcome back. And so kind of gives it away, but obviously this is a ligand-gated ion channel. We used these words before, but now you kind of know what this means. This channel here is not opening in response to depolarization to a threshold. That's what the voltage-gated channels do along the axon, right? These channels open and close based on whether or not the neurotransmitter is present or not present. Okay, that's what we mean by a ligand-gated ion channel. So if we break this terminology down, the ligand is the neurotransmitter, okay, is that little intermediate chemical signal between the neurons. And binding of this neurotransmitter to the protein basically opens its gate. When we're saying ligand gated, meaning that that gate opens or closes based on the ligand. And since this protein in response to the ligand will open and allow ions to flow through, that's what we mean by an ion channel. Okay, so its job is to let ions in, but only when the ligand is bounded to it. Okay, so that's how we change the membrane potential in a graded way. All right, so let's take a closer look at these postsynaptic potentials, these changes in voltage that are caused by these chemical synapses. Okay, so postsynaptic potentials, or PSPs, can either be excitatory or inhibitory, right? They can basically tell the cell you should be more likely to fire, or you should tell it to be less likely to fire. Okay, so let's compare the two different types. So an excitatory postsynaptic potential, which I'll show you in green here, they increase the likelihood of a new action potential being triggered here at the axon hillock. Okay, so here's an example of an excitatory postsynaptic, excitatory neuron, and when it signals the postsynaptic membrane, it causes the membrane potential to go up a bit closer to the threshold. On the other hand, an inhibitory PSP, right? So when we have an inhibitory signal come in, that causes the membrane potential to go down, to become hyperpolarized, making it less likely to initiate an action potential. Okay, so when you have a bunch of excitatory and inhibitory signals coming into this neuron via the dendrites in the cell body, right, synapsing with the dendrites in the cell body, and you were to measure the membrane potential at the axon hillock, you'll find that the membrane potential keeps kind of bumping up and bumping down depending on what signals are coming in and from which neurons. So maybe this one fires and this one sends a signal and this one sends a signal, and then that causes the membrane potential to bump up but not quite hit the threshold, right? And then maybe an inhibitory signal comes in and knocks it back down. Okay, and then maybe nothing happens for a while, so there's green areas, no signals coming in, but the sodium-potassium pump and the potassium leak channels are still bringing the potential back down towards resting. Then we get another excitatory, then another inhibitory, back down where we started. Then we get a couple of excitatory ones, and finally, oh, here we go, we finally hit the threshold at the axon hillock. Those voltage-gated channels are now triggered to open up, and now we have an action potential. Now we can't stop it. Now that action potential is going to start here and spread all the way down and pass on the signal. Okay, so overall, if you add up all of the postsynaptic potentials, right, based on where they are, the types of synapse you have, how much chemical is released, and how close they are to the axon hillock, they're going to add themselves up in different ways at the axon hillock. And if the axon hillock does somehow reach the threshold potential as a sum of all those effects, that's when an action potential gets triggered. Okay, so this is how a neuron can integrate information from multiple sources. I know this has been a long time since that student first asked that question on Tuesday, but when they asked, how do you integrate the information? So it's not as though the cell body is sitting there getting information and writing it down and thinking about it. What it's doing instead is just basically having the membrane potential go up or down based on what signals are coming in and how fast. Okay, so once you integrate all those positive and negative signals, then it can kind of fire or not fire based on how they add up. Okay, so another scientific thinking question for you. Okay, so for this channel here in the postsynaptic membrane, right, what specific type of membrane channel would cause this synapse to produce an inhibitory postsynaptic potential in this postsynaptic membrane? Okay, so when you open this channel, what kind of channel do you want that to be if you want the membrane potential to go down, to be inhibitory? Okay, so here are your options. Pause the video now and make your choice and commit to it on your worksheet. Okay, welcome back. So I would say that it would have to be a ligand-gated potassium channel. Okay, ligand-gated because, again, we're not waiting for the voltage to change in this membrane. We're waiting for the neurotransmitter to open up that channel. And it would have to be a potassium channel because if we want the membrane potential to go down to inhibit an action potential, then we need positive ions to leave. So we need potassium to be able to diffuse out of the membrane protein, out of the postsynaptic cell instead. Okay. So again, if you have any questions about the general structure and function of neurons and how synapses work, please just come by my office hours next week and I'd be happy to talk to you about it. But next, let's move on and kind of go and look at things at a bigger scale now. Let's look at how different kinds of neurons can link together to form the very complex nervous system that we have in our bodies. All right, so the first question I want you to consider is what a nerve is, because you must have heard of nerves before. It's not the Emma Roberts and Dave Franco movie, never heard of it actually, but a nerve, to give you another hint at what a nerve actually is, when you get a pinched nerve, as a physiotherapist might call it, what you might experience is some radiating pain. For example, if you have a pinched nerve here, you might feel pain all the way down your leg. You might also feel numbness. So if you have a pinched nerve in your wrist from carpal tunnel syndrome or something like that, you might feel numb throughout part of your hand. Or you might also get muscle weakness across a relatively large area as well. Like if your sciatic nerve is damaged, you might feel weakness in your knees. Right? So considering that, pause the video and think about what a nerve might actually be. All right, welcome back. So a nerve is actually a bundle of neuron axons. Okay, so those long parts that are sending action potentials across long distances, they're bundled together just to make it a stronger kind of rope-like structure that can handle some a little bit of squeezing and damage. So these axons come from multiple neuron cells, so many nerve cells are bundled together along the axons. And so the cell bodies and dendrites must be located somewhere else then if the nerves only contain the axons. So that's where we get ganglia, okay, or single being ganglion. So a ganglion is basically a cluster of the cell bodies and dendrites of a whole bunch of neurons, and the axons extend away in a nerve usually, okay? So why are ganglia important? Well, if you look at kind of more ancestral looking species like cnidarians, which include things like hydras and jellyfish and such, they don't actually have a brain like we do, right? So instead of having a brain, they basically, their whole nervous system is what we call a nerve net. So it's a bunch of nerves connected together, but you also see these kind of dark spots here, which are the ganglia, where you get these nerves actually interacting with each other, where they might integrate some sensory information. And based on how that sensory information is integrated, it can then signal the correct response to those sensations. Now, what happened as animals evolved to have bilateral symmetry more and more, so meaning kind of having a head and a tail end, and even more so as cephalization evolved, meaning that the concentration of sensory organs near the front head end of the animal, and more like kind of the digestive functions and reproductive functions further down in the animal, what we found was that more and more of these ganglia get combined, right? So the ones involved in integrating like the antennal information and eyes and all that kind of stuff, they were combined together to form increasingly complex brains and spinal cords. So that's what became the central nervous system that we see in humans and a lot of vertebrates today. Now, the peripheral nervous system then is all those other nerves and ganglia that are found outside of the central nervous system. So central nervous system is in red here, the brain and spinal cord, and the peripheral nervous system is all these green and blue nerves heading out from the CNS. Okay, so now that we're looking at this kind of full nervous system level, we can still kind of look at individual neurons and look at how they function within these complex systems. So basically make a good connection between individual nerves, as you studied them so far, and these larger nervous systems, right? Because the structure of each nerve really much reflects their specific functions. All right? So overall, sensory neurons, their job is to carry signals from your sensory receptors, for example, the light receptors in your eyes, and sensory neurons will take information from the sensory receptors and send that information to the central nervous system. Okay, so sensory neurons from all over your body sending signals to your spinal cord and brain. Now, once it gets to your central nervous system, a whole bunch of interneurons will take that information and integrate and process these signals that came from the sensory neurons. Right? Then these interneurons can then connect to motor neurons, and the motor neuron's job is to then send signals to your muscles and to other parts of your body to react to whatever's going on inside and outside of your bodies. Okay, so as a scientific thinking question as well as a practice exam question, what I want you to do is kind of draw upon what you know so far about nerves and nervous systems, and also just use your sense of logic and critical thinking in order to answer this question. All right, so here we have kind of neuron A, B, and C, and these are very generalized structures for the three types of neurons that we pointed out earlier. So one of these three is a generalized sensory neuron, one of them is an interneuron, and one of them is a motor neuron. Okay, so what I want you to do is figure out which one of these three is a sensory neuron, and then once you've done that, try to justify your answer. Okay, so use your worksheet for this, and as a bit of a tip, often when you're trying to justify your answer or explain your answer, it's easier to explain why the other two choices are incorrect than it is to tell us why your choice is correct. So if there's only three options, tell me why the other two don't make sense, and the remaining one will be very well justified in that way. All right, so pause the video now and try to figure out which of these three is a sensory neuron and explain why. Okay, welcome back. So let's do this kind of one step at a time. So let's look at this general neuron A first and ask yourself whether you think that neuron A is likely to be a sensory neuron. All right, so pause and think about this. Okay, welcome back. So this is unlikely to be a sensory neuron because if you think about it, sensory neurons generally have to send signals a very long distance, right? So maybe not so much from the eye to the brain, but for example, if you were sending a signal from your fingertip where you might be touching something hot to your spinal cord, we're talking about almost a meter in length. So one, you should see, instead of just a whole bunch of kind of short dendrites and axons, you should see a very long dendrite or axon to carry that signal a long distance, right? And since that signal has to be traveling a very long distance, you want that signal to travel quickly, especially if you're burning your finger here. And so you would expect to see myelination as well. Now, we didn't include myelination in any of those three options, but that's still something to look for in other figures. All right. So myelination, you'd expect to get faster transmission of these signals when they have to go such a long distance. Okay. So considering those are some reasons why this is not a sensory neuron, what can A be then? Okay. So take a close look at the structure and tell me, is this then an interneuron or a motor neuron? Okay. And once you've decided that, write a justification. So use your worksheets to write down your reasons for thinking that this is either an interneuron or a motor neuron. Okay. So pause the video now and fill out your worksheet based on that. Okay, and welcome back. So I would say that this is an interneuron, right? Because if you think about it, sensory and motor neurons, both of them will need to have long dendrites and axons, right? Because if you're sending a signal from your fingertip to your brain, that needs a long dendrite or axon. If you're then sending a signal from your brain back to your finger to move it out of the way, that needs another long dendrite or axon as well. So for this example neuron here, we don't have either, right? We don't have long dendrite or axon, right? So it shouldn't be a motor neuron or a sensory neuron. Also, it makes sense that a generalized interneuron should have lots and lots of branches for both the black dendrites here and the red axons here. So lots and lots of branches for both. Because if you think about it, an interneuron is made to receive and send information from and to many other neurons, right? So to give you an example, it's kind of a made up very simplified example. In your central nervous system, so say somewhere in your brain, some excitatory postsynaptic potentials, so some kind of exciting signals, can come in from a bunch of different sensory neurons, and those sensory neurons can be carrying information from a variety of other receptors. So for example, this interneuron may end up receiving signals that originated from your eye, that if you later integrate it would tell you that there's pie on the table. So that would give you some information about the sight. Maybe there'll be other sensory neurons sending information from your nose to confirm that, okay, things smell like pie. And as you put it in your mouth, there might be other sensory neurons coming from the taste receptors of your tongue, telling the same neuron that, okay, that tastes like pie, we have pie. Also, touch can be involved as well, as you feel the warm, yummy crust and everything like that. All right, so all of these senses, the signals from all these sensory neurons coming in to meet with these different branches of the dendrites can kind of stimulate parts of your brain that help you to recognize when pie is present, right? So having received these signals from many different sensory neurons, you could then also use this interneuron to link on to many other interneurons as well. All right, so you might trigger a bunch of responses. For example, some synapses from this axon terminus to another interneuron might activate your speech center so that you can speak and ask for more pie. I like pie, by the way, you might have noticed. It might also signal from this branch to some of the motor neurons to get you to go and grab some more pie from the table. It might also activate some other interneurons deeper in your brain, in other parts of your brain, that will make you think about past pies so you can compare, which ones are better and that kind of thing, right? So, I mean, this is kind of a silly made-up example, but notice for an interneuron that's supposed to send and receive a whole bunch of very complex signals to process and make decisions, it makes sense to have lots of branches to link with many other interneurons. Okay, so again, notice that structure reflects the function. All right, so we've eliminated option A, right? So let's look at option C and think about, is this a sensory neuron or not, and why? All right, so pause the video now, and using kind of the ideas and concepts you just learned, think critically about whether or not this is a sensory neuron. Okay, go ahead and pause. Okay, welcome back. So remember that when you receive information on all these different dendrites, you can basically integrate a whole bunch of information, but then once that information is integrated, all you can send out from this cell body is an action potential, right? So that action potential could be firing at different frequencies. You can maybe fire an action potential slowly, like once every 2 seconds, or you can fire it way more often than that. But pretty much that's the only signal you can send, because you have one axon leading away from this cell body, and all that you can send along the axon are these all or nothing action potentials that all look exactly the same. So all you can do is fire slowly, fire more rapidly, or not fire at all. That's all the information you can send along the axon. So if you think about it, if you actually had a sensory neuron that branched out this much and received information from a whole bunch of sensory receptors, for example, say from a whole bunch of light receptors from your eye, how the heck is this sensory neuron supposed to integrate so much detailed information along just one axon, firing at different frequencies, right? It's pretty much going to be impossible, right? There's no way that a cell receiving from so many light receptors is going to be able to pass that information onto your brain, right? Unless somehow it can kind of boop, 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 kind of like spell it out in Morse code and describe what you're seeing, right? So unless you can Morse code your way to saying, you are beep, 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 looking at a beep, 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 yellow HB pencil, blah, 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 all right? So that's not going to happen. So it makes more sense that this is not a sensory neuron, this is a motor neuron, right? So again, think about structure and function. Why does it make sense that a motor neuron will look like this? We'll have so many branches here, but just a few branches at the axon termini. Okay, so why does the structure make sense? Okay, so pause the video now and answer these questions in your worksheet. Okay, welcome back. So a motor neuron, think about it, it's once it activates and sends a signal along its axon, it will just reach a few muscle fibers within one of your muscles. So it only tells a few of the muscle fibers to contract, so you can control how many of those fibers are contracting at once. Okay, so that makes sense that there are very few branches at the axon termini. But there are a lot of reasons why a particular set of muscle fibers should contract or relax, right? Which is why it makes sense to have these long branch dendrites to receive all these excitatory and inhibitory signals from many other interneurons that might help you decide whether you should be contracting or not. So, for example, some sources of inhibitory signals could be things like the fact that you're tired, right? If you're feeling really tired, drained, lacking oxygen in your blood, that might send some inhibitory signals to this muscle fiber, so you're less likely to contract it. Also, if you're feeling pain from damage to your muscles, that's another signal that may reach this motor neuron and inhibit more contraction. And so this inhibition would cause this muscle to relax. On the other hand, in terms of excitatory postsynaptic potentials, some other things can cause the muscle to still contract despite being tired or being in pain. For example, if you have a strong strength of will and you're very determined to lift that weight, like my friend Ruthie here, you may still be able to push past the pain and tiredness and still send strong enough excitatory signals to still contract the muscles. Or maybe you have some sort of parental instinct. Let's say your kid just crawls underneath your arm. You're not going to drop the dumbbell on them, so you would again say, okay, forget the pain, forget the tiredness, I'm going to hold this dumbbell up a little bit longer. All right, so overall what I'm meaning to convey here is that it makes sense to have all these branch dendrites to receive information and signals from many different interneurons in order to integrate that signal and decide whether and how much to contract these muscle fibers. Okay. Okay. So now that we've kind of looked at interneurons and motor neurons, let's finally reveal the answer. So B is the sensory neuron, right? Notice that this one has just a few dendrites and a few axon termini. Okay. So pause the video in a second now, and I want you to use your worksheet again to write about why this structure makes sense for the sensory neuron. Okay, so pause the video now. Okay, welcome back. So it makes sense that it has just a few dendrites because, like I said, you can only integrate a little bit of information and pass it along the axon through action potential, so no point in having too many branches. So it uses a few dendrites to receive information from just a few sensory receptors, so maybe a few touch receptors in your skin. So if you're touching that area, these ones get activated, you send some signals to the brain to say, oh, I'm being touched right here. Okay? And it also makes sense that when it reaches the axon termini that you don't have too many branches, because generally speaking, you're just going to be activating a few interneurons, right? But that can still kind of activate many parts of your brain because those interneurons then move on to activate a whole bunch of other interneurons from there. Okay, so those interneurons can connect to basically millions of other interneurons by branching out and touching many others. So within the CNS, this bit of information will get spread to all parts of the brain and spinal cord. Okay, so after the brain senses that, oh, you're being touched at the tip of your nose or something like that, and that information is processed through many different things, that might stimulate some of your motor neurons to sweep away whatever's touching your nose, that kind of thing. Okay, so that can stimulate the specific response based on all the excitatory and inhibitory signals being sent to the motor neuron. Okay, so that kind of gives you the three kinds of neurons found in the nervous system and kind of why they are structured the way they are. So again, if you have any questions, come see me next week. So moving on. So now that we've connected neurons to your nervous system, let's get a kind of bigger, broader overview of the vertebrate nervous system as a whole, right? So there's 4 different levels with which we can subdivide and kind of split the nervous system into smaller groups. So we're going to explore each of these subdivisions together. So the first division is this. So if you take your entire nervous systems, all these nerves and glial cells in your body, you can divide them into the central nervous system, which is shown in this red box up here, and the peripheral nervous system, which is everything down here. Okay, so the central nervous system, or the CNS, is made up of the brain and the spinal cord. Right? So it's shown in red here, brain, spinal cord. And the peripheral nervous system, or the PNS, is made up of the nerves leading to and from the central nervous system. So it doesn't include the central nervous system, but it includes all these nerves that are kind of sending information to the CNS and sending information away from it. Okay. It also includes not just the nerves, but any associated ganglia that are located outside of the CNS as well. All right. So that's the first division, nervous system divided into CNS and PNS. Second division, we can take our peripheral nervous system and divide that into 2 parts as well, which is the afferent and the efferent divisions of the peripheral nervous system. So the afferent nerves are the ones that send information to the central nervous system, and the efferent nerves are the ones that send the information away from the central nervous system, right? So these signals could be sent from the brain, for example, to different effector organs, so different muscles and different glands, right? So to give you an example to help you remember this, an example of some afferent nerves could be ones that carry information from your eyes to your CNS, right? So let's say that you're watching Batman v Superman and you see Ben Affleck being Batman. That's going to kind of send signals through the afferent division of your peripheral nervous system to your brain. And after it's been processed, then you might send some information, some signals out through your efferent nerves towards, say, your facial muscles or your tear ducts, and signal all the different things that make us effing cry. And that kind of shows how the efferent division would affect different parts of your body due to signals from your CNS. So to help you remember, Affleck, the sight of him is sent via afferent nerves to your brain. and then the efferent system will send all the signals out that make you F and cry. Okay, F, F. Okay. So moving on to the next division. So if you take the efferent division, so all those nerves are coming away from the central nervous system, you can divide them into two parts as well, the somatic and the autonomic nervous system. So green for somatic, blue for autonomic. All right. So your autonomic nervous system is involved in regulating most of your internal organs and glands and smooth muscle, basically most of the parts of your body that require nervous system control, but control without us being conscious of them. So pretty much these are things that are working automatically. So for example, if you need to control blood flow to specific muscles or specific organs, that's done automatically through your autonomic nervous system. Same with digestion. You don't have to think about digesting. It's just being controlled automatically by your nervous system on the autonomic side. Your somatic nervous system is the part that controls your skeletal muscles. So skeletal muscles move when you tell them to, when you think about it consciously. So these are voluntary movements caused by signals sent through your somatic nervous system. Now, depending on the textbook, including our own, often the somatic nervous system, they also include the afferent nerves. So the afferent nerves that come from our skin, our sensory organs, our eyes, ears, mouth, that kind of thing, all the senses that we're conscious of, we think of that as part of the somatic nervous system as well. So somatic nervous system, you can think of it as all of the conscious senses coming into the brain, as well as the conscious movements that you send out. Okay. So a bit of a scientific thinking question that I'll use to help you kind of take this terminology and apply it to a new context so that you kind of understand the terms better before we move on to the final division. All right, so here we see a picture of Bruce Lee blocking a kick from Kareem Abdul-Jabbar, who's a very famous basketball player as well as a very good student of Bruce Lee's. And this is in the movie Game of Death. So question for you to consider is, which part or parts of the nervous system were directly involved in Bruce Lee detecting the kick? Okay, so here are your options. More than one of these can be correct. So in your worksheet, write down the letter of all of the parts that you think are directly involved in Bruce detecting the kick. All right, so pause the video and write down your answer now. Okay, welcome back. So the afferent division is definitely involved because we're talking about senses being sent to the central nervous system. So afferent division is involved. And the afferent division is part of the peripheral nervous system, so we can't leave that out either. So that you can say the peripheral nervous system in general is involved in detecting things. You can include the somatic nervous system as well, because even though it's kind of shown as part of the efferent division here, some textbooks do talk about senses like what you see and what you hear being part of the somatic nervous system. Also, you can say the central nervous system as well, because yes, there might be signals from your eyes and ears telling you that a kick is coming, but these nerves, sensory nerves themselves, don't recognize what a kick is. It's only once your central nervous system processes that visual information that you can say that Bruce actually detected it as being a kick, right? So so the bold ones are kind of obvious answers. These other ones are ones that you can argue for being true or untrue, depending on how good your argument is. Okay. All right. Second question. So what part of the nervous system are directly involved in Bruce moving his hand to block the kick? So we're talking about taking action now. Okay, so again, pause your video and select all the correct answers and think about how you would justify each of your choices. Okay, pause the video now. Okay, welcome back. So again, there's some obvious kind of true answers, such as the somatic nervous system. So if we're sending signals away from the brain to take action, the somatic nervous system is definitely involved because we're moving our skeletal muscles to make the block happen. We also are looking at the efferent division because, again, anytime we're sending signals out of the CNS towards muscles or glands that are taking action, we're looking at the efferent division. Also, those are all part of the peripheral nervous system, so we can't leave that out either. So these are all obvious good answers. The central nervous system, you can also say, is involved because you wouldn't have the movement unless the central nervous system is coordinating and sending information out. So that's not the best answer, but one you can argue for being true too. All right. So a few more scientific thinking questions, and these are again good examples of questions that I could ask you in an exam, the kinds of questions where you have to really think, okay, and explain things. So when people ask why Bruce Lee was such an amazing fighter, a lot of people would just say, oh, he has really good reflexes. He just like blocks everything so fast. He's got really good reflexes. So as a biologist, would you agree with this statement or not? And why or why not? Okay, so pause the video and just make the best answer you can and the best justification you can to this question, and then come back and we'll discuss it. Okay, so pause the video now. All right, welcome back. So as a biologist myself, I would not agree with this statement because as biologists, we define reflex in a very specific and different way from the general public. So we would describe a reflex as an unconscious, involuntary response. So for example, if your hand happened to be touching something hot and you weren't paying attention, that would send a signal to your spinal cord, and without even sending anything to your brain at all or sending anything back from the brain, it basically, your sensory neuron connects to an interneuron, which pretty much directly connects to your motor neuron for your bicep and triceps, telling them to move your hand away from the hot thing. All right? So basically, these reflexes are mediated by interneurons in your spinal cord, and it basically bypasses the brain altogether. So you do not kind of think about the fact that your hand is hurting. You do not send the signal to your bicep. All of that is mediated by your spinal cord. Okay? So in other words, the reflexes that biologists talk about, we're born with. Okay, this is not something you can learn. You can't learn to have an unconscious reflex in the biological sense, right? So why is it that Bruce is able to, block attacks faster than most other people when he was alive? Well, it's through learning, right? It's because he practiced, had lots of experience with sparring and fighting, and therefore learned ways and trained his body and brain to respond faster. Okay, so as an example, let's look at someone who is not at all Bruce Lee. So let's say that someone's blocking a kick for the first time. All right, so there's a lot of different steps that are necessary to go from something happening to your body responding to it. First of all, some external stimuli are coming in. For example, just the fact that the foot is moving closer and there's light reflecting off of it that you can detect. So when the stimuli are coming in, you need sensory receptors that can detect that stimulus. So maybe in your eyes, you'll have receptors that will catch the glint of the sweat coming off the foot, and then you can see the foot is coming. So the sensors at least sense the light from the foot. And then your afferent sensory neurons, your afferent neurons, will take the signals from the sensory receptors and send it to the brain, and in a non-specific way basically say that something's coming, like there's the light coming off of something, and you're sensing that light and kind of telling the brain that something's happening. All right. So as these signals arrive to the central nervous system, that's where we really have to start processing things. So that's where your central nervous system processes the visual information and says, okay, we got a kick coming. It's the right hand leg. So this is the right leg. So if it's the right leg, the foot will be coming from the left side, but my left hand is down here, so I got to move it up there, and I got this much time to get there. When I get there, I got to make sure the wrist is straight and the elbow is bent, otherwise I might break one of those two, etc., etc., etc. There's a lot of things your brain needs to think about the first time around to make sure you do a block properly. Right? So once that processing is done, you might finally then use your somatic nervous system, which is part of the efferent division of your central nervous system, and signal your muscles to move. So essentially now sending signals out to your muscles telling, okay, hey muscles, time to move that arm quick before you get kicked. All right? Now, unfortunately, if this is your first time blocking a kick, you are probably going to be too slow. By the time the kick comes and you move your hand, you already would have gotten hit in the face. That's how you end up looking like this. This is me after getting kicked in the face during a martial arts class. So definitely need more practice. So how does practice change your nervous system in a way that makes it more efficient or better at doing things with experience? Okay, so how does practice affect the nervous system, and how does learning happen? Well, in general, the changes that happen to your neurons and your central nervous system in response to practice, in response to experience, is called synaptic plasticity. So plasticity meaning kind of changeability, flexibility in our synapses. So generally speaking, when you use a synapse more frequently, when you use one nerve cell to signal another cell, those synapses get strengthened. Whereas those that you don't use very often are weakened or they're lost altogether. That synapse is gone, okay, or that at least doesn't have a strong effect. So to give an example of how that works, long-term potentiation is one way in which a synapse that's used more often starts to have a stronger effect, right? So if you take a synapse and you use it often, so you send signals through that synapse often, the postsynaptic potential, so the change in voltage of the receiving cell, becomes bigger and bigger, right? So whereas one action potential might have produced a small reaction before, it's going to produce a bigger reaction later. So when you're summing up the positive and negative, the excitatory and inhibitory signals from different synapses, the one that you use more often is going to give you a much bigger increase or decrease in the membrane potential compared to one that's used less often. Okay, so how does that actually work? How is it that experience can make this synapse have a bigger effect on the membrane potential? Well, one way is by making it so that every action potential releases more neurotransmitter. The more neurotransmitter you release, the more it stays in this cleft, activating your membrane channels in the postsynaptic membrane. Another way is by adding more of these channels instead in the postsynaptic membrane, so by adding more ligand-gated channels. So that way when you release the neurotransmitter, there's more channels opening and there's more ions moving in or out of the postsynaptic membrane. Okay? So that's how long-term potentiation can work. Another way in which we can see synaptic plasticity is through synaptic pruning. So if you don't use a synapse often enough, or you don't use it for a long time, then sometimes the synapses are lost. So here we have a synapse between one neuron and another, and if you don't use it often enough, then that connection can actually be lost. All right, that's kind of why I need to review student names a lot when I'm trying to learn them, right? Because if you don't review it, if you don't use it, you lose it essentially when it comes to memories. Now, myelination is another way in which learning can change your effectiveness, your nervous system. So as you fire a neuron more and more, right? These Schwann cells actually grow more and more to wrap around the axon and make it more efficient, right? So more myelin is added to kind of commonly used neurons, and that causes that neuron to carry the signal much faster than before. So if this is for blocking kicks, this is very, very helpful. This also kind of points out why myelination is important and why when someone has multiple sclerosis, as one of my best friends was recently diagnosed with, MS is basically a really horrible disease, genetic disorder where the body's immune system starts to attack these Schwann cells that make the myelin. And as you lose this insulation, the coordination of the firing and carrying of the signal gets messed up, and you experience a lot of very bad symptoms as your nerves no longer fire in the coordinated way that it should. All right, so myelination is very important, but it's also part of the way in which experience changes how your nervous system works. All right, so the result of all these different types of synaptic plasticity and changes in myelination is that you can basically speed up or strengthen the impulses or the synaptic connections that are needed to go from detecting to giving a coordinated response. So basically, all those steps we talked about that go from the kick is coming to blocking the kick, you can make all of those connections, all of those steps faster and more efficient the more you practice it. And if you practice it enough times, then it becomes what we call muscle memory. So even though muscles can't memorize anything at all, right, what we're talking about is all those connections that we talked about from sight to blocking can become more and more efficient with practice. So you can almost think of it as every time you practice a certain response and action, you're walking the same path in the woods again. And the more you take that same route over and over again, it becomes faster and more clear, it becomes easier, and becomes also less conscious each time as well. Eventually you can walk your way home without even thinking about it. You can block a kick without really consciously thinking about it much either. Okay? So all of this is talking about not a biological reflex, but a programming of your central nervous system such that when it does receive at your brain all these sensory stimuli, it knows what to do with it more and more quickly because you've done it many times before. All right? So I think all this stuff is really, really fascinating, really cool. But then if you think about how this might work across the number of nerve cells that we have, like this is just a very small piece and a few small bits of understanding in a very, very complex and fascinating puzzle. Because if you think about it, we have around 100 billion neurons, and they're connecting to each other about 100 trillion times, so 100 trillion synapses. So yeah, what we understand from one synapse has to be extrapolated a lot for us to fully understand the nervous system. All right, so if you watch the 2-minute neuroplasticity video that's posted on the website, you'll be able, sorry, on your reading guide, you can explore the idea of learning a bit further. The link to this video and your focus questions are in your learning guide. Okay, so use those focus questions to make sure you focus on the right parts of that video. All right. So, next question for you to think about is, considering how complex the brain is and all the connections and how plastic it is, how it's always constantly changing, will we ever understand the mechanisms underlying kind of our consciousness, our intelligence, our ability to feel emotions, to learn, and to remember things? It seems kind of so complex it's out of reach, but seriously, we are trying, right? There are scientists out there working on what we call the Human Connectome Project, which, according to their own project kind of mission statement, it's a project that's meant to construct a map of the complete structural and functional neural connections in vivo, meaning in a living organism within and across individuals. So we want to know in one individual and what the differences are between individuals in terms of all the connections that you find inside a brain and how they act in different actions and senses. All right, so it used to be that getting to know every neuron, every nucleotide in our DNA was out of reach, but we had the Human Genome Project, and now we have a lot of information and a lot of ways to use it. Human Connectome Project is the next step, looking at neuron function rather than DNA. All right, so let's continue and finish off our different divisions of the nervous system now. All right, so we've divided CNS nervous system into CNS and PNS, the PNS into afferent and efferent divisions, and then efferent division divided between the somatic nervous system, which we just talked about in detail, the senses and the motor kind of responses that we're conscious of. So now let's take a closer look at the autonomic nervous system, the side that takes care of your body unconsciously mostly, and we divide that into the sympathetic and parasympathetic nervous systems, right? So both the parasympathetic and sympathetic nervous systems are involuntary, and they're pretty much both always actively signaling, but they kind of do opposite things, all right? So one might be more active than the other depending on the situation. So the parasympathetic nervous system is more active when we're in a safe place, when there's not much danger around. So when there's not much danger around, this side of the nervous system is going to kind of signal things that help you do more resting and digesting and having more sex as well. Okay, so all the things you do when you're safe. The sympathetic nervous system is more active when you are in danger. All right, so the way you can remember these two terms is the parasympathetic system, parasympathetic nervous system is what you might activate when you have a parachute and you're nice and safe and you're floating kind of safely down to the ground. The sympathetic nervous system, when you don't have the para, is what would happen if you're falling without the parachute. So if you feel like you're going to die, that's when your sympathetic nervous system is more active. Okay, so here's a figure that shows you kind of what the parasympathetic and sympathetic nervous system activates when they're working. So when the parasympathetic nerves are firing kind of more actively than the sympathetic ones, you'll be doing a lot of things related to rest. So you'll let your pupils constrict a bit because you don't need as much sensory information. You'll salivate and digest things in your stomach and have more movement of your intestines as well. So all your digestive stuff will be activated. And also you're not going to bother beating your heart or breathing as hard. Okay. You also can, pee when you need to, and you're more likely to have erect genitals for having sex. Now, if you are in danger, if you're falling from the sky and you're going to die, or you're being chased by a lion, then your sympathetic nervous system will become way more active, and you'll notice that the responses are kind of the opposite of the parasympathetic nervous system. So you will dilate your pupils so you can kind of see what's going on around you more quickly. You will inhibit your salivary glands, your stomach, and your intestines, and make it so that you have less digestion happening, because it's more important to get away from the lion that's chasing you than it is to digest the meal that you just had. All right. You also relax the bladder so you're not trying to pee at the same time, and so on and so forth. Okay. So generally speaking, both the parasympathetic and sympathetic nervous systems are both actively signaling all the time. But when you are very safe, then you'll have more of your parasympathetic nervous system firing and less of your sympathetic, whereas if you're in danger, it's the other way around. All right, so it's the balance between the activity of the two systems that determines how your body's acting in any particular point in time. All right, so As your next scientific thinking question, here's a practice exam question you can kind of try to practice with at home. So if you put yourself into one of these two scenarios, either being eaten by a polar bear or eating a bowl of ice cream, describe all the ways in which your body would respond to that situation, right? So use proper terminology to refer to all the relevant parts of your nervous system, and also talk about things in terms of what stimuli are coming in, how it's processed by the central nervous system, and then which effector organs your central nervous system might signal in order to help you respond to it, right? And also in terms of your sympathetic nervous system, how might they change in response to your situation as well. Okay, so that's a good practice question to try at home. All right, so one last topic to cover is our really big and complex and beautiful brain. So we've learned a few things about how different parts of the brain work and what the brain does. A lot of it early on from damages to the brain. So for example, if somebody was behaving in a weird way and hearing voices, after that person dies, we might dissect the brain to find, oh, this part of the brain was really damaged by an accident, and that might be why he was hearing voices, right? So that's kind of like the simple old school way that we might have learned how, like what a part of the brain does. So for example, if someone damaged the cerebellum and suddenly they're very uncoordinated and having trouble walking and standing up straight, then that let us know that maybe the cerebellum is useful for that. Nowadays, we also use a lot of brain scans to look at what areas are active when we're doing different activities as well. So that's a less invasive way to figure things out. So some examples. So what have we learned? We've learned that there are 4 distinct structures in our brain, each of which have very different functions. We've also learned that looking at one of those parts, the cerebral cortex, each region is specialized for doing something different. Now you don't have to memorize what all these regions are and where they're located, so do not try to memorize this figure. That's something you'll learn if you become a brain surgeon, but you don't need to know it right now. Okay? But you should kind of understand how we made this little map, right? So one of the ways we can do it is to put someone in a PET scan, right, and get them to do different activities. So this is what the PET scan looks like when someone is hearing different words, and this is what happens when they're seeing different words, when they're reading the words instead. It's quite a different part of the brain being activated. Here we have what happens when someone's speaking words, and that's why that's one of the ways we located the Broca's area. And also if we're generating words and thinking about them, it's in a different part as well. Okay, so PET scans basically, the way we light up different parts of the brain is by detecting changes in the blood oxygen levels, right? So whatever area is active, they're using up more oxygen, there's more blood flow, and therefore the tracer we injected into the blood shows up more, right? So a pretty simple but cool system. So notice when you look at these different figures that in general, the left side of the brain controls the right side of the body. Okay, that's one thing. So you see, if this is kind of the top view of a brain, this is the left hemisphere. All of these parts that control sensation and movement of the right side of the body is found on the left side of the brain. So also, when you look at your sense of touch and temperature, so the somatosensory cortex, which is what's shown in blue in this picture here and here, it's located right beside the motor cortex. So it kind of makes sense that your senses and your movements in response to those senses are kind of located close together. But not only are those two slices located side by side, you also find that where you sense information from your legs and your lower body, for example, in your somatosensory cortex, it's actually located right beside the motor cortex for your legs and knee and hips and so forth as well, right? So it kind of makes a kind of sense that you're going to be integrating information between your senses of one area and your movements in that area, right? Also notice when you look at these pictures that things are kind of stretched in a weird shape, in that there's not a lot of kind of brain tissue dedicated to information coming from, say, your hips or your knees and that kind of thing, because those aren't very sensitive parts of your body, they're not important parts. But then you look at your fingers and thumbs or your lips, there's way more, because there's a lot of nerve endings there, because you need to know a lot of information about what you're eating or who you're kissing based on that sensory information. So that kind of gives you some hints for the scientific thinking question here, which was for you to think about why it is that you dedicate kind of more brain tissue to one part of the body more than the other. You will explore this in more detail in lab next week. All right. So quick thing to remember is that you don't need to memorize any of these regions. Do not spend any time trying to memorize, which part of this little fold controls the fingers and so forth. Okay, so that's pretty much it for this lecture. So just to go over your learning objectives. So now that you've gone through both lectures and once you've reviewed these lectures in your textbook, you should be able to describe the structure and function of a neuron in a lot of detail, and all the details that we've given in class. You should also be able to describe how an action potential is initiated and produced, and how it also propagates along a neuron, and also how signals are passed from one neuron to the next. So there's a lot of details in all of those steps that you should understand well. You should also be able to distinguish the functioning of each major part of your central and peripheral nervous systems using proper terminology, and also kind of predict what's going to happen and which parts in a specific kind of interesting situation. You should also be able to talk a bit about how the nervous system itself changes when you learn things or when you repeat certain experiences. Okay, so that's pretty much it for this video. Thank you so much for taking the time to read it, to listen to it, and I will see you all in class next week. Thank you very much.